Hi, this is Mrs. Murdock, and this is a post-lab discussion of the Bacterial Transformation Lab. So at this point, you should have your 4 Petri dishes in front of you, and hopefully you saw some really cool results on those, and that's what I'm here to talk about. So what you should have is, if you lay the Petri dish out the same way that I am, it'll help you to trace what I'm doing. So let's put your LB that had no plasmid right here. And then your LB that had ampicillin in it, ampicillin in it, that also had no plasmid. We'll put that right there. Then we'll put the LB, the Luria broth, with ampicillin antibiotic over here that did have plasmid given to it. And then we'll put the LB Luria broth with ampicillin and also arabinose sugar over here that also had plasmid given to it. Okay, so what you did in your lab was you had 2 samples of bacteria, of live bacteria, and the negative sample didn't get exposed to a plasmid, like if I were to draw a little piece of DNA over here, and this plasmid had a couple of genes on it that were going to help it have new abilities. So the plasmid had a gene for antibiotic resistance. Antibiotic resistance. There was a gene for that on the plasmid. And also a gene for pGlo, which makes the bacteria glow green when you put UV light on it, which you're going to try in a minute. Right, so this is the plasmid, right, which you can't see really, but that's what it has on it. It has these genes, one for antibiotic resistance and one for pGlo to make it glow green. And the negative ones, that was the sample of bacteria that you didn't try to transform. You didn't expose it to the plasmids. So what should have happened is with just Luria broth in there, LB stands for Luria broth, you should have had the entire surface of the agar should be covered with bacteria. This is called a lawn. So it should all be just milky white, off-white bacteria covering that, because there's nothing to stop the bacteria from growing. There's no antibiotic in there. There's just Luria broth that it likes. So this plate it's growing because there's no antibiotic to stop it, and this shows that your bacteria, the bacteria sample that we got from the laboratory, is competent, right? It was competent. It was able to grow. It did just fine in ideal conditions, right? You gave it food, you gave it warmth, you didn't put anything in there that was going to kill it, so it grew just fine, right? Okay. So this plate, on the other hand, should have no growth at all. There should be not a single dot of bacteria on here because you put ampicillin in here and you did not give it the genes for antibiotic resistance that could protect it. So there should be no growth at all on this plate. Right? And this shows, hopefully you can predict this before I say it, this shows that the ampicillin worked. It shows that the antibiotic, or the ampicillin, the antibiotic that Mrs. Murdock put into the agar itself, when she poured that agar, it had the antibiotic swimming around in there. You try to grow bacteria on the surface and it can't grow, it dies because the antibiotic kills it. Okay, so that shows that our antibiotic was working. Now over here, the second sample of bacteria that you were working with, you actually went over to Mrs. Schultz or Ms. Rankin or me and got a little syringe full of plasmid, that clear liquid drop that went down in there, and then you heat shocked that. And what we hope is that those living bacteria took the plasmid, the little piece of DNA, took it inside themselves and gained the ability to resist the antibiotic. But they're not all going to be able to do it. Transformation efficiency is usually really not that high. It's maybe never even reaches 10%. So what you'll see on this is you'll see colonies. You won't see a lawn like you did over here. You'll see individual colonies of bacteria that we're able to transform. We're able to take the plasmid inside themselves and successfully use the gene on the plasmid to resist the antibiotic, right? So this shows, right, this plate shows that transformation is successful, right? That some of the bacteria, not all of them, were able to transform and resist that antibiotic and resist the ampicillin, the antibiotic that was in the agar that Mrs. Murdock put there. Right. Now that also means, and this could come as a question, is that every single one of the cells that's growing on the surface here are nothing but transformed bacteria. There aren't going to be any bacteria who are able to grow on this that didn't take the plasmid in. So remember that. 100% of the bacteria on this plate took that plasmid in, are transformed. Okay? Over here, now you have this funny thing called ERA, and ERA is short for arabinose. Arabinose is a sugar. See the ose at the end? Lots of sugars end in ose. It's just a sugar. So again, you should see about the same pattern. You should see individual colonies. Once again, you had bacteria. You were able to transform some of it, not all of it, and those are the colonies that you will see. Right? Here's the difference. This also shows transformed colonies, right? Transformed colonies, transformed bacteria that were able to resist the antibiotic. But here's the thing. When you have those little LED lights, those UV lights, the little handheld things that you wave across the surface of the bacteria, and you're looking for that pGlo gene to wake up. You're looking for that bright green color. And what happens is this. So, get some nice green going here. Here's what happens. These guys all glow green when you put that light on them. You get that light on these. They cooperate and they light up green when you put the light on, right? These do not. So these guys, they don't glow. They don't have that lovely iridescent light when you put the UV light on them. These do. They do glow. Now, why is that? That's funny, because we didn't give a different plasmid to this one than we did to that one. They both got the exact same plasmid. Here it is. That plasmid has 2 genes on it. One of them is the antibiotic resistance gene. So that's why some of them were able to grow at all. And the second is the pGlo gene, right? Which makes them glow green. But it didn't do that on this plate. These guys are not glowing, whereas these guys are. And the difference is the arabinose, the sugar. Now, why would that be? So think about the lac operon. Remember, the lac operon was something that bacteria have, and that when they're in the presence of milk sugar, lactose acts as an inducer and makes things happen, right? That's the way that the lac operon wakes up and starts making its genes. Well, arabinose happens to be an inducer as well. So if you go upstream from the pGlo gene up here, I'm not going to be drawing this in great detail, but imagine that there is an operator right there, and it has a repressor on it, suppressing, preventing the pGlo gene from being made to make those glowing green chemicals, right? But if arabinose shows up just like lactose, it connects to that repressor and allows the pGlo gene to be expressed. That's why the presence of the sugar is causing those bacteria to glow green. And it's the only bacteria that are going to glow green. These other ones over here, the ones that weren't transformed, also aren't going to do it because they didn't get the plasmid at all. So all of these things are the things that you need to remember. This is testable stuff, okay? Next Tuesday, for my class, you're going to get a paired essay that's going to ask you questions about these results, and you need to be able to explain yourself. So in addition to this nice kind of conclusions that you have here, I just want to show you another image that you might see on a test question, or that you might see maybe in the essay we'll see. So this is a very similar experiment, just about the same experiment, except there's no arabinose here, there's no green glowing gene here. The only thing they did here was introduce an ampicillin resistance gene. So if we look at this, what's going on here is here you have wild type E. coli, meaning they were not transformed. So these would be your negative plates, negative and negative. And here, these E. coli on these plates 3 and 4 had plasmid added to them. So they would be your plus plates, right here and here. All right, so with no ampicillin in the agar, remember we're not dealing with the pGlo gene here at all. We're just dealing with whether the bacteria grew or not, whether the ampicillin was there or not. So with no ampicillin, both plates grew just fine, lawns, right? They grew and grew and grew, whether they were transformed or not. With no ampicillin and nothing but luria broth, right? That's all these guys had was luria broth. They had food and no ampicillin to stop them, so they grew and grew. Whereas with the ampicillin over here, you should be able to predict, even if you're not shown a picture, there's no growth when you have no transformation, no gene given to resist the ampicillin, but you have ampicillin here in the plate. So this would be an LB amp plate, right? Amp, sorry, I can't spell. An LB amp plate. So there's ampicillin in this agar, that wild type E. coli that was not transformed, negative, no plasma, can't grow. Then over here you see, here you have the same thing. You have an LB amp plate with ampicillin in the agar, and you have but you gave this guy a plasmid with an antibiotic resistance gene. So now you have a little bit of growth. See that? Some of the bacteria are able to grow. So here's a question they'll try to trick you with. Okay, I'm going to try to help you with this. So what if you wanted to only have bacteria that were antibiotic resistant? Which of these plates would you go to get bacteria that were exclusively antibiotic resistant? Which one? 1, 2, 3, or 4? If you said this one, you are correct. Because this plate only has colonies on it that were isolated, right? And were successfully engineered to take in the plasmid and have that resistance. So only plate 4 has 100% transformed colonies. Now, what if you wanted bacteria that were 100% not engineered? Which plate would you go to? You knew all of them, 100% of them had not been engineered. 1, 2, 3, or 4. In that case, 1 would be the answer. 1 would only have bacteria in it that did not get a plasmid and thus were exclusively wild type. None of these guys can survive antibiotic, but if that's what you want, there it is. So what does that mean about this plate here? Plate number 3, think carefully. Plate number 3 has both non-transformed and transformed bacteria in it. It would be very difficult to separate them. They're all in there together, growing successfully, because there's no ampicillin to stop them whether they've been transformed or not. Okay, so that's the kind of thing, those are the kind of conclusions and analysis you should be able to do on the paired essay that will be asked about this. And that's what bacterial transformation is about. It's kind of a really exciting field. I hope you found the lab interesting and fun, and I hope that's helpful.